Plant tissue culture, PTC, is a collection of techniques used to maintain or grow plant cells, tissues, or organs in a sterile environment. This environment is typically provided by a controlled laboratory setting. Types of plant tissue culture. 1. Meristem culture. Involves the culture of meristematic tissues, which are undifferentiated cells that have the ability to differentiate into different tissues and organs. 2. Callus culture. Involves the culture of callus tissue, which is a mass of undifferentiated cells that can differentiate into different tissues and organs. 3. Organ culture. Involves the culture of entire organs, such as roots, stems, or leaves. 4. Protoplast culture. Involves the culture of isolated plant cells without their cell walls. Steps involved in plant tissue culture. 1. Selection of explant. The part of the plant to be used for tissue culture is selected, such as a leaf, stem, or root. 2. Surface sterilization. The explant is sterilized to remove any microorganisms that may be present. 3. Initiation of culture. The explant is placed in a sterile culture medium, such as agar or liquid medium. 4. Incubation. The culture is incubated in a controlled environment, such as a growth chamber or greenhouse. 5. Subculturing. The culture is subcultured regularly to maintain its viability and prevent contamination. Applications of plant tissue culture. 1. Micropropagation. Plant tissue culture is used for the large-scale production of plants, such as orchids and potatoes. 2. Plant breeding. Plant tissue culture is used to produce hybrid plants with desirable traits. 3. Production of secondary metabolites. Plant tissue culture is used to produce secondary metabolites, such as alkaloids and glycosides, which have medicinal properties. 4. Conservation of endangered species. Plant tissue culture is used to conserve endangered plant species. Advantages of plant tissue culture. 1. Rapid multiplication. Plant tissue culture allows for the rapid multiplication of plants. 2. Disease-free plants. Plant tissue culture allows for the production of disease-free plants. 3. Uniformity plant tissue culture allows for the production of uniform plants. 4. Year-round production. Plant tissue culture allows for the production of plants year-round, regardless of the season. Limitations of plant tissue culture. 1. High cost. Plant tissue culture is a labor-intensive and expensive process. 2. Limited success rate. Not all plant species can be successfully cultured using plant tissue culture techniques. 3. Risk of contamination.